Hi, welcome to the Bio and AD Tutoring Center videos. The topic of today's video is enzymes. So basically, what is an enzyme? An enzyme is a catalyst or something that speeds up a reaction. So in previous videos, we talked about reactions being spontaneous or non-spontaneous depending on their delta G, but whether a reaction is spontaneous or not has nothing to do with how fast the reaction occurs. And so there's a lot of reactions or metabolic processes in our bodies that are spontaneous reactions. They actually have a net release of energy, but they will happen so slowly that without enzymes, we really wouldn't be able to survive. So something really important to note, though, is that enzymes lower the energy of activation. They do not change the delta G. So the energy of activation is just the energy needed to get a reaction started or going, whereas the delta G is the net change in free energy. So right here we have this graph, and it's showing the free energy versus the progress of the reaction. So the black indicates the reaction without an enzyme, and the red indicates the reaction with an enzyme. So you notice for both reactions that delta G is the same. If this is our reactants and this is our products, the energy of our reactants versus the energy of our products is the same no matter what, if we have an enzyme or not an enzyme. What changes is the energy of activation, or the energy needed to get over this hump to proceed with the reaction. Or in other words, to get up to the transition state. So just always keep this in mind, because a lot of people will forget this important fact, and it will trip them up. It's also important that we just know some basic terms associated with enzymes. So if this is our enzyme right here, usually we have a substrate in the active site. So the substrate is the specific molecule that binds to the enzyme or the active site, and then the enzyme will change it somehow or break it apart or add different parts to it. And so there's this idea of induced fit when it comes to enzymes, and induced fit just says that enzymes will change their shape slightly to fit substrates better. So they won't change a lot, but they'll alter their shape a little bit to fit things better. So then that brings us to, how do enzymes lower the energy of activation? So that's what they do, but how do they do that? And so there's four main ways that they do this, and depending on the reaction, the enzyme might do this multiple of these or just one of these. It really just depends. But the first way they do it is they bind substrates in the proper orientation. So a lot of times what we're trying to do in a reaction, we're trying to bring two reactants together to form one product, and they have to meet together in the right way to do that. And so, for example, if we have this blue and this black molecule, and they're trying to come together, and this little black part is trying to meet up with that blue part, if they're just flying around randomly, not flying, but moving around randomly in whatever space they're in, the likelihood of them meeting in that proper way to form the bonds they need to is very low. But if you add an enzyme in, then the enzyme will bind each of them in the proper way, and so that then they can react the way they need to. So that just speeds up the reaction. Another thing that they do is they strain bonds, so it favors the transition state. So say, for example, we have this substrate and we want to break it apart into two different molecules by breaking a bond right here. If we put it in this enzyme, the substrate has to bend more to fit in the enzyme, and as it bends, that strains that bond, so it eventually can break into our two different products. And that's just one example of how it can strain bonds. And then other things that enzymes do to lower activation energy is they change the local environment. So some reactants need to be in certain environments, acidic or basic, and so it can do that. And then last, it participates directly in the reaction, so it breaks and forms bonds. And so there's just lots of different examples of that happening. So that's just the four ways that enzymes lower the activation energy. And then enzymes also are considered, not enzymes, but an important thing to know about is cofactors. And cofactors are just non-protein helpers. So they just help enzymes in different ways. And we generally think of two types. There's metal ions and coenzymes. So metal ions are just anything on the metal side of the periodic table. And they a lot of times help with like electron transferring. And then coenzymes are just organic cofactors. So a common example is vitamins. And as you talk about cellular respiration and photosynthesis, there's going to be some really important coenzymes that assist in those processes, and they're called, one of them is NAD plus or NADH when it's reduced. But you'll get into more of that later. And so the last two things I just want to touch on is saturation and naming. So saturation is just the idea that your enzymes can only go, or your metabolic process can only go as many enzymes you have. So if you have a bunch of reactants that need to be changed into products, but you only have a couple of enzymes, and then all the enzymes you get full are saturated with those reactants, and so your process can only go as fast as those can take in reactants and shoot out products. And the last thing is naming. It's important to know how we name enzymes. So typically, the majority of enzymes end in ASE, ACE. So some examples are DNA polymerase or kinases. And so the second part of it usually tells you what the enzyme is doing, and the first part usually tells what it's doing it to, so what it's forming or using to form that thing. So polymerases typically synthesize new molecules, monomers to polymers. That's where the polymer comes. So the polymerase, DNA polymerase, is making DNA, synthesizing DNA polymers from monomers. And then kinases, we're going to talk a lot more about those later, but kinases are enzymes that add phosphates to different things. That's all for today's video. In the next video, I get more into how we regulate enzymes. Thank you for watching, and I hope it was helpful.